Hi, today I'm going to talk about my PhD, which is about the evolution of cytoplasmic incompatibility mechanisms in the Wolbachia Culex pipiens system. I'm supervised by Mylène Weill and Mathieu Sicard. So first, Wolbachia are maternally transmitted endosymbiotic bacteria, which infect 40 to 60% of arthropod species, which is quite a pandemic. And it does a lot of weird things, such as feminization, which is transforming genotypic males into fully functional phenotypic females, male killing, changes from male-producing parthenogenesis to female-producing parthenogenesis, and also what we're going to talk about today, which is CI. So the main idea in CI is that whenever an infected male here is crossed with an uninfected female, this cross will be sterile, so no progeny. Whenever an infected male is crossed with a female that bears what is called an incompatible bacteria, then the cross will be sterile as well. There's been some theoretical models of CI. The main model now is what is called modification rescue model. The idea is that whenever an individual is infected, Wolbachia will have modified the sperm, which is what is represented here by those purple dots. And so in case of fertilization, if the fertilization occurs with an infected egg, there will be some embryonic defaults resulting in the embryonic death. However, when the egg is infected, which is represented here by those yellow dots, there'll be a rescue by Wolbachia and the development will be normal. Some genes implied in this have been found 3 years ago, so quite recently, and they're called CytA and CytB in the Wolbachia that is infecting pipiens, so WPIP. The reason why we're studying WPIP is that it does quite a lot of interesting things. So the main idea is that there's been 5 groups of WPIP described so far that have been named WPIP1 to WPIP5. And if you cross hosts infected by WPIP from a given group, most of those crosses will be compatible. While if you cross individuals infected by WPIP from different groups, in most cases, it will be incompatible. So that led to the conclusion that several factors are required to explain such complex CI patterns. And indeed, what Manon Bonneau, so previous PhD student, found was that the seed genes that I mentioned earlier were amplified and diversified in WPIP. There can be up to 6 or 7 copies of a given gene within a single genome. An interesting thing as well is that they correlate with the CI patterns. So what I've been doing so far is studying the diversity and evolution of those genes with the question of how we can get such a huge diversity in those genes. I've been studying particularly the intragenic architecture of those genes because as you might see here, they seem to be made of a lot of recombination blocks. So I'm identifying them and trying to see whether some correlate or not with the CI patterns. And I've also been trying to see whether there's a phylogenetic signals in all those what we call variants, because if they explain CI, we would expect that. What wasn't expecting is that there's no phylogenetic signal, so we're trying to see something else, so maybe geography or maybe having a look at more like smaller blocks. I've also been working on the phenotypic variations of CI and their links with the seed genes. So we are looking at some temporal variations of repertoires, which are the full set of variants within a given genome, and trying to see whether those repertoire changes correlate with some phenotypic changes. And I'm also working on the gene expression to see whether all those genes that are present are expressed or not, to see what it means to have so many genes. Another thing that I've been working on is on phages. The reason is that those genes have been located in prophage regions of the genome. And so we're trying to see whether they can be found in lytic phages, because if so, they could be a means of horizontal transfer of the genes. And so, yeah, we've been isolating lytic phages and trying to have a look at the lytic phage diversity and whether or not we could find the genes within the phage. Well, I think it's going to be time. Thanks for your time. And if you have questions, please don't hesitate.